Hello everyone, my name is Mahmoud Rani Rahman. I have been doing research on COVID-19 for a year now, and I think this topic really needs some attention. So here we will be talking about how indoor weather or indoor climate plays a huge role in COVID-19. I and along with my group, they work really very hard. We try to figure out exactly what indoor climate plays a COVID role, especially in USA. So we conducted a kind of statistical analysis on the COVID-19 cases. I'll be talking about more on this. I hope you will pay attention because we are very sure that this study will gonna be very helpful to understand the COVID dynamics. Let's move on. Okay, let's talk about the biology first. So when virus goes in our body, for example, when we inhale air, virus can goes into our lungs or in the nasal airway, then it goes into the cell. It goes into the cell in two ways. One is called the endogene fusion process, another is called cell membrane fusion process. Endogene fusion process is basically a two-step process. It is first packaged, it becomes encapsulated by a packet within the cell membrane, and then it enters as a packet inside the cell, and it waits to further fusion process. And cell membrane fusion process is basically a faster fusion process because involves one-step process. And we need to remember here is that the important point is that membrane fusion is not really a spontaneous process, rather is a regulated process. What I mean by regulated is that it needs a certain construction, certain solution like pH or membrane arrangement, and so if this construction met, then the fusion process can happen. That's why I would argue that cell membrane fusion is a faster process and the endogene fusion is a slower process. And I'd also like to draw your attention here for the HIV virus. As far as I know, the HIV virus cannot penetrate, cannot go through the cell membrane fusion process, it stops. But it can go into the cell through endogene fusion process, and probably that is the one of the reason that HIV virus kills people very slowly. So for respiratory viruses, or in general respiratory viruses, or maybe for the COVID virus, I would like to argue that the cell membrane fusion is the most efficient fusion process that happening in our body, and that can makes us sick. When we get virus, generally virus replicates faster in our upper respiratory tract compared to the lungs or other body parts. So we need to really carefully examine what's going on in our nasal airway. So I do research on fluid dynamics, and I have been doing research on drop coalitions for more than 5-6 years. So I know very well how and what plays role in drop fusion process, and I think the same thing can happen in virus fusion process. So what can happen is that when there is a membrane tension difference between virus membrane and cell membrane, then there is a critical membrane tension for which the viral RNA can be released into the cell further away from cell membrane. And for other differences, say the cell membrane tension and virus membrane tension are very similar, then or maybe the cell membrane fusion, virus membrane fusion is much higher compared to the cell membrane. I'm talking about the tension, membrane tension. then the viral RNA will release to the cell, but it will reside near to the cell membrane. So there is a huge difference between cell membrane is released further away from cell membrane and cell membrane is released next to the cell membrane. When sorry, I'm talking about the viral RNA released in the cell membrane. So when viral RNA reside next to the cell membrane, this is my assumption is that, and from the experiment of the colloidal hydrodynamics, my PhD is basically on colloidal hydrodynamics, and I know that there is a hydrodynamic interaction between molecules and the wall. So when RNA reside next to the cell membrane, it is mostly unlikely that RNA can go further away anytime soon, because it will stay there because of this hydrodynamic interaction. So either of the two things can happen. This RNA can be destroyed by the other protein, or the virus can form almost similar virus through budding process. So that means one virus coming in and one virus going out, so it's not replicating. But when virus is further away from cell membrane, then the cell membrane, the replication machinery, or I believe this is a ribosome, that can make another copy of the RNA, and that can replicate the virus. This is the way of virus replication process. So I'm basically talking about the cell membrane fusion process because I think this is the faster process, and if this replication happens due to cell membrane fusion process, then someone can get sick quite faster way. Now we need to know, as I said, this can happen only when there is a critical membrane tension difference between cell membrane and the virus membrane. Now we need to look at when this can happen. So the tension of on the virus membrane can happen when the temperature drops. So say the virus is attached to the cell, but it's not fusing, but when temperature drops, then the construction on the virus membrane, the virus becomes shrink. As it is shrinking, at some point the shrinking is stopped, and then the membrane tension develops. But the same thing can happen for the cell, but as cell is much larger than the virus, the temperature may not, slight changes of temperature may not affect the virus cell membrane that much, but it can affect the virus membrane, because virus is much smaller, and when it shrinks, the membrane tension difference, and at some point there could have a critical membrane tension difference between cell and virus, and at that particular moment viral RNA will be released to the cell interior further away from cell membrane, and this is the deadliest way of virus infection. We already wrote an article explaining all these details maybe more than 5 months ago, and we explained what could be the cause. So now we need to focus exactly how our nasal airway temperature can be modulated, and that I will talk in the next slide, and I will draw your attention about the weather and air properties. Let's move on. In the previous slide, I talked about the critical membrane tension difference that can lead to virus infection, like deadliest type virus infection. So critical membrane tension, that means membrane tension can be modulated by different ways. For example, the surrounding pH of the surrounding liquid can play a role here. Maybe cholesterol can play a role here, or there may have other mechanism, but temperature also plays a role here. I already discussed a little bit that when temperature drops, virus shrinks, and at some point the shrinking process is stopped and membrane tension develops. The less temperature is, the higher membrane tension of the virus. So, but there is a critical membrane tension difference. That means there is a critical temperature at which virus replication will be much higher. That's why we need to look at how temperature can be dropped in our nasal airway. So I think it's very obvious that the air we inhale can reduce our reduce the temperature of our nasal airway. Now we need to understand a little bit about the heat transfer mechanism of our nose. So our nasal airway is basically a 100% or close to 100% humid environment. So once we get air, then we generally get air from the environment, the atmosphere. So if we get air from, say, if we get air as a dry, then the dry air goes inside our nasal airway, and the dry air becomes fully moist, and this moisture is coming from the nasal airway liquid regime. So it's kind of wet, and this nasal airway water can evaporate into the upcoming air, and through this evaporation process heat is lost and your nasal airway becomes further cold. That means temperature and humidity both plays a role here to modulate your nasal airway temperature. And temperature and humidity, the exact mechanism that I told you can be explained through wet bulb temperature. Now I need to explain a little bit about the wet bulb temperature. Wet bulb temperature is, say you got a piece of cloth, you make it wet with water, and then you wring extra liquid from it, and you leave it for a drying. You leave it in your room to dry. Then the liquid in that cloth, the water will evaporate. Depending on your room temperature and humidity, the evaporation process will be different. So that means the heat is lost, and if you put a thermometer on the wet cloth, you will get a temperature that is called wet bulb temperature. And wet bulb temperature is always lower than your room temperature. If your room is 100% humid, then wet bulb temperature and room temperature will be same. Now this wet bulb temperature, this like the cloth temperature, can define the wet bulb temperature. At the same way, it can define the nasal airway temperature. Now the nasal airway temperature is different than the wet bulb temperature of the room, but it is easily understood that we can correlate these two parameters. That means if the wet bulb temperature is higher, our nasal airway temperature will also be higher. If the wet bulb temperature is lower, our nasal airway temperature will also be lower. And also how faster you are taking air, that means breathing rate can play also in the dynamics. So there are a complexity here, but the point here is that as there is a critical membrane tension, there must have a critical temperature, and as there is a critical temperature of the nasal airway, there must have a critical wet bulb temperature. That's what I am showing here in the results section, in this result. So what I did, I studied all the US counties. I got the weather data from NOAA website, National Aeronautical, I forgot the full name, anyway, so we got this weather and we interpolated weather data, interpolated indoor wet bulb temperature from outside weather. So I have explained in detail in our preprint. I'll share the link here, and so if you go through this preprint, you will understand how exactly we got this indoor wet bulb temperature, even though we did not physically measure indoor wet bulb temperature for every home. So there may have slight error, but it mostly I am very confident that we did the right job here, and the temperature we measured is very much close. And so what I did, I took all the temperature at different time frame that you can find in my in our preprint, and from all those data, so many data points, then we got those temperature, indoor wet bulb temperature, which did not vary significantly for any 9 days period. So for 9 days, that means say February 1 to February 9. For these days, I took all the weather data and I got the wet bulb temperature, and I figured out the mean wet bulb temperature and the standard deviation. When standard deviation is much smaller, that means for that 9 days period, this weather was, the indoor wet bulb temperature was almost constant. So on the left plot, I only showed those temperature which was constant for those 9 days period of time. And I took the COVID data for next 9 days period, because when somebody is getting sick, he takes, he is being tested, then it takes some time. So I took a 9 days grace period as a sort of infection time and reporting time. So I took weather data for 9 days and I took COVID data for next 9 days. And what we got here is really interesting, and I think this is really important plot here on the left. You can see there is a clear indication that there is a critical wet bulb temperature, and I can say with confidence that when temperature is higher than 14 degrees Celsius, indoor wet bulb temperature is higher than 14 degrees Celsius, I did not see any county that reported more than 100 cases per 100K population. So this is really significant. But when temperature is say 12, 13 in that range, you see number of cases average, I think almost 1,000, and in some of the cases it goes more than 1,300, 1,500 cases per 100K people. So definitely this plot shows that there is a critical wet bulb temperature at which virus replicates much faster. If we can stay in an environment, or if we can inhale air that has wet bulb temperature is higher than 14 degrees Celsius, we may reduce the number of cases significantly. And also you need to pay attention here is that above 14 degrees Celsius, the number of cases is lower than 100, but still it's not a zero. That means there may have other dynamics, or in statistics we couldn't figure it out. But in most of the cases, a significant reduce can be possible if we can raise indoor wet bulb temperature. And here on the right two plots, I am showing here that at two different time frame, at two different places in the United States, you can see that number of cases was much higher for 13 degrees Celsius and 11.5 degrees Celsius. And this is what I think is that may relate to the virus strain. So each virus strain has different membrane construction, may have different spike protein, and it may respond temperature slightly differently. So for each virus strain, there may have different critical temperature. And from this plot, we see two temperature really stand out than rest of them, and number of cases is much higher. Okay, let's move on to the next slide. Now let's talk about the mask. So we need to really understand how masks play a role in this pandemic dynamics. To my understanding is that mask really cannot block virus sufficiently to reduce COVID cases. But it helps in other ways that I will explain later. The reason I'm talking because I cited at least 2 articles. If you consider those studies, then you will understand that virus really, even though if you wear masks, you will get virus. If you wear N95 category masks, there is a chance that you may get a small amount of virus. And once you get a small amount of virus, it make a colony of virus by replicating. So I'm very confident that mask is not playing a role by blocking virus, rather it's playing a role by keeping your nasal airway warm. As I have discussed that there is a critical temperature, there is a critical wet bulb temperature at which virus replicates faster. That means when you wear mask, what can happen is that a part of your exhaled air you are breathing in, and your exhaled air is moist, almost 100%. So you are taking moist air and also like hot air. That means you are sort of sitting in a room where the room wet bulb temperature is higher, but you are taking your own breathing, and you are taking hot and part of hot and moist air, so you are keeping your nasal airway warm. That means you are avoiding that critical temperature range. That means here mask is helping. But here is another point. If you stay in the colder air, then that means mask wearing can increase the chances that you are getting a critical wet bulb temperature at which you will get sick. That means mask helps, mask makes things worse. So we need to be really very careful about mask wearing. So here I wanted to show you some data here. So the blue dot on the left plot, you can see the blue dot shows the data for December 3 to 11, and this standard deviation is 0.8. That means the temperature, indoor wet bulb temperature, did not vary significantly. And if you look at the next 9 days weather and COVID data, as shown by the red dot, you can see that at around 11 degrees Celsius, the number of cases dropped significantly. So that means cases risen at 11 degrees Celsius, but next 9 days it drops significantly. And the explanation here is that the standard deviation less than 0.8 of it basically shows for those areas where outdoor was really significantly cold. That is shown on the right, top right plot. You can see the red dot shows the outdoor wet bulb temperature. It shows clearly outdoor is very cold. That means when there is a cold for a significant period of time, even though indoor is virus infectious, you are not getting sick anymore because it's a kind of lockdown effect. So in 9 days period, maybe all people are kind of sick, and there is no any people you get to make them sick. But and also when you go outside is much colder. That means you are not making other people sick. Outside is much colder, means you are not replicating your virus, so it is virus unfavorable, so cases is being reduced. So for a longer period of cold significantly freezing weather for a longer period of time makes the number of cases much lower. But for that same time period, if you look at the green dot, when the indoor wet bulb temperature was fluctuating a little higher, like more than 1.2, that happened in certain zone that is shown by the green dot in the top right plot. And those zone, you see the outdoor temperature is basically 0 to 5. Average outdoor temperature is 0 to 5 degree Celsius, outdoor wet bulb temperature. So that means for the next 9 days period, case did not get reduced, rather it goes up, way up, that is more than 1,000. So one part of the region gets number of cases much lower, like almost 200 per 100k people, and another part number of cases goes much higher, like more than 1,000 per number of per 100k people. So the difference is very obvious and very significant. So where these cases rise, the outdoor weather is where the average outdoor weather is 0 to 5 degrees Celsius. And what potentially could happen is that part of the region, people kept wearing masks outside, because widespread we are talking that we need to wear masks when we meet people. So people keeping wearing masks outdoor. That means they are raising nasal airway temperature to the critical zone, and when they are indoor, they are not really wearing masks. It's my observation that people do not wear masks when they are indoor, like at their home. I believe people do not wear masks. Even when they are in the office, if he is in a single, he is doing office, maybe not in a crowded atmosphere, he generally do not wear masks, but when he meet people, he wear masks. So my point here is that you are in a virus favorable indoor and you are not wearing masks. That means your nasal airway in the critical zone, and when you are going outside, when the outdoor temperature, average temperature is 0 to 5 degrees Celsius, you keep wearing mask and you are making nasal airway critical again. So that means you are always kind of critical virus favorable environment. So mask wearing outdoor makes things much worse for that particular time and particular places. Now, why I'm pretty sure is that because I did the same study for during springtime, winter springtime, the earliest time of the COVID, and I didn't observe the same phenomenon. I'm not going through in detail, but you can read our preprint and you will understand what I'm talking here. So we need to understand how mask plays a role here, then we can probably reduce number of cases. Okay, so based on my study that I explained earlier, this is my suggestion, because the COVID pandemic is rising now. Cases are being rising, is rising around the world. So this is a kind of common sense understanding. It's not, I'm not suggesting this from a thorough scientific study, but this common sense understanding is coming from the study I have done previously. So taking shower with hot water for a longer period of time, say for 30 minutes, definitely can help, because when you are taking shower in a enclosed environment and with hot water, you are raising wet bulb temperature significantly. And I measured wet bulb temperature several times, that I found it's like 19 degrees Celsius or so. So it's definitely be helpful. So if you take shower with hot water for 30 minutes every single day, the chances of getting COVID will be significantly lower. Now, I explained about the mask uses. So from that explanation, I would suggest to keep wearing mask indoor as much as you can, and moderate outdoor. Moderate outdoor means if you see the similar outdoor that you live in indoor. In that case, you can keep wearing mask outdoor as well. But if you see significantly cold, like freezing cold outdoor, please avoid wearing mask. So when you are sleeping, you can cover your whole body with your blanket. It will help is that your moisture will be retained in that enclosed environment, and you can raise your wet bulb temperature, and that can help to reduce the virus replication, and perhaps the body immune system can take care of the rest of the virus, and you may not get so much sick. Regular cooking can be another good idea to break the virus favorable critical condition, because you are in a perhaps you will be in a moist and hot environment. Regular indoor exercise can also help keep nasal airway warm, because when we do exercise, we release heat through our respiration, and I explained in our preprint that definitely can help. So when our respiration rate is higher, then our body generates heat and it is lost through the respiration, and that's why our nasal air temperature can go higher. But if you do exercise in cold outdoor, there may have something, for example, you can inhale at a faster rate. So there is a complex dynamics of heat transfer, because you are taking, as your breathing rate is higher, you are taking air at faster rate. So body generates heat, but you are getting higher volume of cold air. So it may help, may not help. I cannot be very sure, but indoor exercise, most likely it will help you. And if it's possible, please sleep in a small room with multiple people, maybe with your children or with your spouse, in a small room or in a small apartment. That definitely helps, because it will raise your wet bulb temperature. And here I showed two plots. You can see the green, I think the purple line and the blue line. The purple line shows the indoor wet bulb temperature for a small room, and the blue line shows the data for bigger room. The blue line indicates that, the purple line indicates that actual wet bulb temperature is much higher than just simply, but actual wet bulb temperature is higher than if you live in a small, in a big room. Okay, so this was my talk here today. I hope this will help to make you to keep yourself healthy, and I hope whoever listen this video will share this knowledge to others. And I strongly seek help from the scientific community. Please review our article. We actually submitted to several top journals. Unfortunately, we got rejected, and we are still trying to convince people. I think we couldn't make this point very clear, and this talk is part of that process. If you are a scientist, I would request you to go through our document. Please leave a suggestion for me and our group, and I think this will save the people's life. I'll be looking forward to your feedback. Thank you very much.